Good morning, everyone. Can you all hear me and see the screen properly and clearly? Just tell me yes or no on the chat box. Yes. So today we will be learning about the most important topic of this chapter, neural control and coordination, which will be difficult for most of the students to understand this particular topic, that is generation, conduction and transmission of nerve impulse. So I have prepared a video about the same as well in order to make that concept very clear and easy for each one of you. And we will be drawing mind maps and concept maps to understand the topic very much clear. So listen to the class without even missing a single point today, so you will get this topic perfectly fine and absolutely clear for each one of you, because this is going to be a very important topic from which a lot of question can come for your NEET examination, and questions have already came as well. So let's anyway move on with this topic. Those who have not become a part of our Telegram channel, don't forget to join our Telegram channel at t.me/neat underscore biopoint. Those who are watching the session right now, don't forget to like, share, and subscribe the amazing platform for more updates on biology. So let's get started with this topic, generation and conduction of nerve impulse. Just take out your NCRT. NCRT lines are exactly pasted over here as the slides, along with some extra images have been added in order to make the concept perfectly fine and clear. So all of you, please open up your NCRTs and read the line along with me. So in the last class onwards, we have been learning that neurons, what are neurons? They are the structural and functional unit of nervous system. So from there onwards, we have been studying that they are specialized cells which helps in the conduction of the nerve impulse. So you have to learn that neurons are excitable cells because their membranes are in a polarized state, that is change in the potential. Okay, change in the potential, that is ions will move. Suppose this is the neural membrane, this is the membrane of a neuron. So this will be in a polarized state, so that ions will move from a higher concentration to a lower concentration, or they can move from a from a higher concentration from inside to the outside. So they can move either from outside to inside, or they can move from inside to outside, because the neurons are excitable cells which membranes are in a polarized state. So my question to everybody, do you know why the membrane of a neuron is polarized? So in today's class, we will be learning about how the membrane of a neuron is becoming polarized or change. How is a change in the potential charge difference is created in the surface of a neuron. Just give me thumbs up on the chat box if the thing is clear for each one of you. Yes? So everybody just imagine this is a neural membrane. Just imagine this is going to be a neural membrane. Okay, in this neural membrane, we have different types of ion channels. They can be voltage gated, they can be ligand gated. Okay, there are various types of ion channels. So I will introduce you three important channels that take part in this nerve generation, nerve impulse generation and conduction. Number one, after I draw the diagram, I will just tell you. The diagram may not be perfectly fine and clear. Channel number one, channel number 2, and the channel number 3. Okay, I have drawn three channels on the screen. That is, this is sodium potassium pump. Channel number 1, ion channel, sodium potassium pump. This channel is the sodium channel. So we find the use of all these ion channels in this particular nerve impulse conduction. Okay, and this is going to be what? Alon potassium channels. So these three are the major ion channels that are present on the neural membrane. So different ion channels are present on the neural membrane, and what are them? They are the sodium channel, potassium channel, and the sodium potassium pump. How is actually a nerve impulse is generated? How is going to be a nerve impulse going to be generated? A nerve impulse is generated based on action potential. How is it generated? Based upon action potential. Clear? What is action potential? Difference in the charge across the neural membrane. That is, maybe like this is a neural membrane. On the outside of the neural membrane it will be positive charge, inside of the neural membrane it will be negative charge. So difference in the charge will lead to the formation of an action potential, which is actually the basis of generation and conduction of a nerve impulse. So guys, just listen over here very carefully. Just listen over here very carefully. I will draw a very important diagram over here. Just I'm drawing a diagram. Okay, this is going to be a neural membrane. Okay, this is a neural membrane. Just imagine, what is this? This is going to be a neural membrane. Okay, one sec. Yes, neural membrane. Mainly in this, we will be learning about potassium and about the sodium and potassium ions. So as the sodium and potassium ion concentration varies, there is a change in the charge and finally leads to the formation of an action potential. I will tell you, this is actually, this neuron is right now at rest. What is the condition right now prevailing? This neuron is right now at a resting stage. Okay, this is right now at the resting stage. At the resting stage, inside the neuron, so this is the intracellular. Okay, intracellular, that is inside. Inside. And this is the extracellular. Okay, this is the extracellular or outside. At the resting stage, the point that you must keep in mind is inside the neuron, the potassium ions will be very high. Okay, inside the neuron, the potassium ion concentration will be very high, whereas very low potassium will be present outside. I'll just tell about that. So at rest, inside the neuron, you will find a high concentration of potassium and a low concentration of sodium. So very few sodium ions are only present inside the neuron. Whereas at rest, when you look at the case with outside the neuron, the sodium ions are very much large in number. That is, the number of sodium ions outside the membrane is very high. So you must understand that at rest, inside the neuron, potassium ions is high, sodium ion is less. Outside the neuron, sodium ion is high, potassium ion is less. So I have told you three different ion channels that work during this process. Number one was the sodium channel, number two was the potassium channel, and number three was the sodium potassium pump. Now we are going to have a look at the sodium potassium pump. Okay, sodium potassium pump is actually what I'm going to tell you right now. So listen over here carefully. I'm just erasing some of the ions over here. I'm going to draw. Okay, this is going to be the sodium potassium pump. What is this? This is the sodium potassium pump. Am I clear? Just give me hearts on the screen or tell me yes or no on the chat box. Sodium potassium pump. So this sodium potassium pump is one of the ion channel that is active at the resting stage as well. So this is active at the resting stage as well. Clear? What do you mean by this resting stage? Resting stage means there is no signal. What is resting stage? Resting stage means there is no signal. The neuron is not having any kind of signals. Clear? So even at that resting stage, sodium potassium pump is active. So what is this function of the sodium potassium pump? The sodium potassium pump will pump 3 sodium outwards. I already told you. And they will pump 2 potassium inwards. So they will pump 2 potassium inwards and 3 sodium outwards. So where is the charge less? So actually what is happening? 3 positive charges are transferred outside, whereas only 2 positive charges are pumped inside. So this one, oh my god. So this particular charge as an extra positive charge outside. So simultaneously when this process continues to happen, just imagine what will be the condition prevailing over here. What will be the condition? Positive charge, this extra bit of positive charge will keeps on accumulating outside the neural membrane or outside the neuron in the extracellular fluid. What happens? The positive charge will keeps on accumulating. What is the condition inside? Only 2 positive charges per, and there is a deficit of positive charge. So deficit of positive charge, you can call them as negative, right? So there is a deficit of positive charge. So opposite is, it will be negative. So you can call them, the inner side of the neural membrane will develop a negative charge. So what happened over here? There become a charge difference. What happened? Due to the activity of the sodium potassium pump, a charge difference is created. Clear? The outside membrane becomes positive charge and the inside membrane becomes negatively charged. This is your point number 1. So the negative charge inside, you can call them, or that is actually minus 70 millivolt. What is it? Minus 70 millivolt. This minus 70 millivolt is known as the resting membrane potential, RMP. What is it? Resting membrane potential. The potential in the resting membrane of a neuron. That is the meaning. Fine, everybody. Fine, everybody. Fine. So let us go back. When a neuron is not conducting any impulse, the axonal membrane is more permeable to potassium ions and nearly impermeable to sodium ions. So the sodium, 3 sodium is pumped outside and 2 sodium is pumped inside. Clear? So this is actually the condition which is prevailing, guys. Look, outside, this is the outside condition. Outside the neural membrane, potassium ion concentration is very less, whereas inside the sodium is also less. So what happened? The sodium potassium pump will pump 3 sodium outwards and 2 potassium inwards. That is actually the main function happening over here. So this is what I would like to just draw to your kind attention. So this triangular shape over here is actually what potassium, and this is sodium. So at rest, outside the cell, potassium concentration is less, sodium concentration is high. Whereas inside the cell, sodium concentration is less, potassium concentration is high. Is it clear? Okay, so let's move on. So what happened, as I've already told you, when 3 sodium is pumped outside and 2 potassium inwards, that will create a net positive charge outside, and that will create a net negative charge towards the inner side of the membrane. So this diagram also we explained it very clearly, right? So I have already mentioned there are mainly 3 channels, that is sodium ion channel, potassium ion channel, and mechanically gated ion channel, sodium potassium pump. I will right now show with the help of a small video how the sodium potassium pump will work. Clear? So listen very carefully. Listen very carefully over here.
Ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values.
So guys, I have already told you this color that have been indicated over here is actually what sodium. In this particular video, it is the sodium ions, and that have been indicated like this is actually what it is, the potassium. Okay, so what happening right now, this is actually going to be what it is, the sodium potassium pump. So the sodium potassium pump is going to accommodate 3 sodium ions over here. Okay, they are going to accommodate 3 sodium ions over here, and this is going to pass this 3 sodium ions outside the membrane. Similarly, what they will do, they will take out 2 potassium ions, and they will pass this 2 potassium ions towards the inner part. So let's have a look at that video.
This is accomplished primarily
Look
by the use of
how the sodium ions have been fixed out, and the sodium ion is going to get it passed on to the outside with the help of an ATP molecule. Look how the ATP molecule, look, 3 sodium ions are passed outside the membrane. Similarly, 2 potassium ions are going to come inside in this similar way with the help of ATP molecule. Clear? Is the process clear? 2 sodium ions have been passed outside and 2 potassium ions have been passed inside. So that is what we have to study over here. Similarly, the membrane is impermeable to negatively charged proteins present in the axoplasm. Consequently, the axoplasm inside the neuron contains a high concentration of potassium and low concentration of sodium. The fluid outside the axon contains low concentration of K plus and a high concentration of Na plus. Clear? These ionic gradients across the resting membrane are maintained by the active transport of ions by the sodium potassium pump. Active transport. What do you mean by active transport? With the help of ATP molecule. We have seen, right? With the help of ATP molecule. 3 sodium outwards and 2 potassium into the cell. Important point over here. Just mark in your NCERT textbook, that is a previous year question for your NEET examination. So finally, what is actually happening over there? A positive charge is developing outside or in the extracellular fluid, and similarly a negative charge will be created inside. So this negative charge will contribute to a potential of minus 70 millivolt, which is referred to as the resting membrane potential. Clear? Yes. So what is this actually making you clear, or what is this actually trying to convey to you? Minus 70 millivolt of charge is required for the generation of a signal. At the instant when the charge reaches minus 70 millivolt, it is eligible for the generation of a signal. Okay. So just imagine, again I am drawing the pump have already, so just imagine like it have reached, the signal have reached minus 70 millivolt. So at the time of minus 70 millivolt a signal is created. Okay, a signal is created. At the time a signal is created, you will have the another thing over here. Next channel is going to work over here. Which is the next channel that is going to work over here? It is the sodium channel. Which is the channel that is going to act next? It is the sodium channels. So as soon as the signals is acted upon or the signals are generated, the sodium channels get opened. What happened? The sodium channel gets opened. So at the instant when this potential reaches minus 70 millivolt, a signal is generated and the signal will make the sodium channels open. So we have already learned that the sodium concentration is more in the outside membrane, right? We have already learned this, right? The sodium concentration is more in the outside. So what happened when the sodium channels are opened? Sodium channels are open, the sodium will move. So the sodium is in a high potential or high concentration outside and it is in a low concentration inside. So as soon as the sodium channels are open, the sodium ions will start moving inside. The sodium ions will start moving inside. Clear? So that means positive charge is going to enter. Positive charge is entered inside the membrane. So we have already learned the potential as minus 70 millivolt. This minus 70 millivolt changes to plus 30 millivolt. Clear? So the process where the sodium channels are opened and sodium ion move towards inside by creating a signal that is minus 70 millivolt changes into plus 30 millivolt. This process is known as depolarization. Formation of the charge difference was referred to as what? Polarization. That was the first process. I forgot to tell this name over there. So what was polarization? Creation of a signal. That is creation of a potential difference. Polarization. When minus 70 is changed into plus 30, it is called depolarization. Okay, it is referred to as what? Depolarization. Next here we are going to learn about the action of the third channel, that is the yes. So just listen over here. I will just give a brief idea, that is minus 70 millivolt. That will actually create a signal. That signal will pump sodium enter or sodium inwards. That will changes this minus 70 to plus 30 millivolt. Okay, plus 30 millivolt. Next we have the next set, that is the potassium channel. This is going to be the potassium channel. Have you ever seen the olden doors? That is old large doors which are very difficult to close. Even though you try to push it and close, it is very difficult. You need to require a large amount of energy. So the potassium channel is exactly similar to such an old gate which is very difficult to close. Once it is opened, it will remain open for a long time. So this potassium channel is difficult to close. just like an old gate which is difficult to close. So what happens over here? Potassium concentration, as we all know, was high inside as compared to outside. So since this channel is open, the potassium ions start moving outside. Okay, potassium ions start moving outside. So what happened? Even though when the signal reaches minus 70, even when it reaches minus 70, since the potassium channels are difficult to close, minus 70 will changes minus 70 millivolt. It reaches minus 80 millivolt. Finally, when it reaches minus 90 millivolt, the potassium channels are closed. Potassium channels are closed. That is referred to as what? Hyperpolarization. Hyperpolarized state. Clear? So anyway, let's come back to the slides and let's discuss. As a result, the outside surface of the axonal membrane possesses a positive charge. Outside membrane will possess a positive charge. The inner surface becomes negatively charged, and therefore you will call that state as polarized. The electric potential difference across the resting plasma membrane is called the resting potential. You might be curious to know about the mechanism of generation of nerve impulse and its conduction along an axon. When a stimulus is applied at the site on the polarized membrane, the site A, that is the sodium channels, become freely permeable for the transfer of the sodium ions. Clear? We will come to this. This leads to the rapid influx of sodium followed by the reversal of the polarity at the site. Look, the site A, it was sodium will move inward. That will lead to the reversal of the polarity at that side. Positive charge will become inwards and negative charges becoming outwards. The polarity of site A is thus reversed, and that process you will call them as depolarized. Clear? When sodium is pumped inside, the polarity is reversed, so you'll call them as depolarized state. The electric potential difference across the plasma membrane at the site A is called the action potential, which in fact is termed as a nerve impulse. And when the potassium channels are open, again this polarity is reversed. Right, again the polarity is reversed, so you will call that process again to be what? Repolarization or hyperpolarized. Again you will come back to the same polarity again, which finally come back to the resting potential. Clear? So now let's have a look at this entire process in detail. Let's discuss each of this point. First, listen to the video very carefully, then I'll explain it again for you if anybody needs it.
The nerve impulse is the sum of mechanical, chemical, and electrical disturbances created by a stimulus in a neuron. The conduction of the nerve impulse can be divided into two main phases: the resting membrane potential and action membrane potential. The neurons are excitable cells. They may be stimulated by physical, mechanical, chemical, or electrical stimuli. The nerve fiber or axon
This is going to be the
covered
neural
by a
membrane.
Axonal or plasma membrane. The neural membrane has sodium and potassium ion channels.
So mainly we are going to
called
first of all look at the
or
sodium
regulated
channel and
channels.
potassium channel, along with the sodium potassium pump also there.
These channels open or close according to the electric potential across the membrane. The ion channels of the resting membrane are differentially permeable.
So this is what we have learned in the beginning. Okay, we have learned this in the beginning itself. That is, the sodium ions are high outside and the potassium ion is less. Whereas inside the axoplasm or inside the axonal membrane, potassium ions are high and the sodium ions are less as well. Clear? Negatively charged proteins are also present inside. That will contribute to the negatively charged.
To ions at different rates. It is more permeable to potassium ions, almost impermeable to sodium ions, and totally impermeable to the negatively charged proteins of the axoplasm. Therefore, the axoplasm inside the axon contains a high concentration of potassium ions, negatively charged proteins, and a low concentration of sodium ions. On the contrary, the extracellular fluid outside the axon contains a low concentration of potassium and a high concentration of sodium. This differential permeability is maintained by a sodium-potassium pump.
Look, this is going to be the
present inside
sodium-potassium pump, which is going to pass 3 sodium outwards and 2 potassium inwards. I have shown you the video for that. Look, 3 sodium is pumped outside
potassium ions that enter
and
the
2
cell.
potassium is pumped inside.
This electrical potential difference across the membrane in an unexcited nerve fiber is called resting potential, and the neuron is called a polarized nerve fiber. When a stimulus is applied to a site, say
So that
A,
is actually going to be the site number A, where the
polarized
positive
membrane,
charge is outside and the negative charge is
the sodium
inside.
ion channels open.
So
and the
as
membrane
each
at the
process,
site
each sodium
becomes
pump
freely
pumps and
permeable to
potassium
sodium
pumps are
ions.
going to take place, what happened? So at
This leads
definite
to a
position, you
rapid
will have the
influx of
change in
sodium
their
ions
polarities.
that reverses the polarity of the site.
Look, finally the entire polarity has been
That
changed.
is,
So
the
this
outer surface of the
process you will
membrane
call them as the
becomes negatively
depolarization,
charged,
when
and the
the
inner
outside
surface
will become
becomes
negative and inside will
positively
become
charged.
positive, and only the sodium channels are going to be active.
This reversal of polarity across the two sides of the membrane is called depolarization. The electrical potential difference across the plasma membrane at site A is called the action potential. This action potential travels as a wave of depolarization along the length of the nerve fiber in a particular direction and is called the nerve impulse. At site B, the axon membrane has a positive charge on the outer surface and a negative charge on the inner surface. As a result, a current flows on the inner surface from site A to site B. On the outer surface, a current flows from site B to site A to complete the circuit of current flow.
Again, the site A has come to the same position
This
back,
reverses the
positive
polarity at the
outside and
site.
negative inside. Similarly, this process will
Action potential
continue
is
like that.
generated at site B. Thus, the impulse generated at site A arrives at site B.
Then that will be converted to the next one. Then again, finally the entire, it will pass through the entire neuron. Clear?
At the peak of action potential, the permeability of the membrane to sodium ions decreases, while it becomes more permeable to potassium ions. This is because sodium channels start closing and potassium channels start opening. However, this part of the membrane regains its original polarity within milliseconds, and this phenomenon is called repolarization. A repolarized nerve fiber undergoes a refractory period of a few milliseconds, during which the original ionic distribution is restored by a sodium-potassium pump, which actively transports sodium ions outward and potassium ions inward. This returns the membrane to its resting potential, and the neuron is ready to receive another stimulus. A junction helps transmit the nerve impulse from one neuron to another. These junctions are called synapses.
So we already studied this, right? In the last class, I have mentioned this about the synapse, which is a junction between two adjacent neurons is called what synapse. We'll learn about this in today's class.
A synapse is formed by the membranes of a presynaptic neuron and a postsynaptic neuron.
Presynaptic neuron means the topper end. Post means the beginning of the next neuron. This is called what? The synaptic cleft. And what all other synaptic knob contains acetylcholine, which is actually a neurotransmitter.
Which may or may not be separated by a gap called the synaptic cleft. There are two types of synapses.
So we'll come back to over this chemical synapse and the electrical synapse after we discuss the topics in detail. Okay, so let's first of all come back to the topic. Okay, so I think you have got the conduction and generation of a nerve impulse. Yes. So a nerve impulse is transmitted from one neuron to another neuron through junctions called synapse. A synapse is formed by the membrane of a presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap called the synaptic cleft. There are mainly two types of synapse. One, electrical synapse. Two is the chemical synapse. At electrical synapse, the membrane of pre and postsynaptic neurons are in very close proximity. Okay, they are very close proximity. I will show you after this. Electrical current can flow from one neuron to the other neuron through these synapses. Transmission of an impulse across an electrical synapse is very similar to impulse conduction along a single axon. And it is always faster. Electrical impulse is very faster than a chemical synapse, but electrical synapses are very rare in our system. They are very rare in our system. So which type of synapse is more common? It is the chemical synapse is more common in us. Clear? At chemical synapse, the membrane of the pre and the postsynaptic neuron are separated by a fluid called the synaptic cleft. Do you know how the presynaptic and post-synaptic neuron transmit an impulse? There are certain chemicals called neurotransmitters. The vesicles are filled with these neurotransmitters. One of its example is the acetylcholine. Okay, in the last class, I have just given you a brief idea about the same. Acetylcholine is a neurotransmitter. I've shown you the image also. So this is where neurotransmitters are present. Clear? Neurotransmitters are present over there at the vesicle. Clear, everybody? So this released the neurotransmitter while bind to the specific receptors present on the postsynaptic membrane, which allow the entry of ions and will generate a new potential, which can be excitatory or inhibitory as well. Clear? So before we end over there, I will show you the rest of this video as well before we end. Yes, we'll watch from the synapse onwards.
And a postsynaptic neuron, which may or may not be separated by a gap called the synaptic cleft. There are two types of synapses on the basis of the nature of...
Look, chemical synapse, there is a synaptic cleft. But in electrical synapse, there is no cleft. They are very close proximity. They are in very close proximity.
These are chemical synapses and electrical synapses. Chemical synapses are common in the human system.
So chemical synapse is very common in the human system, where a presynaptic neuron and a postsynaptic neuron is separated by a fluid-like membrane called synaptic cleft.
A presynaptic neuron.
And now you doubt how will they be transferred. So the synaptic vesicle will contain synaptic vesicle, which contains neurotransmitter such as acetylcholine.
Each synaptic vesicle contains neurotransmitter chemical molecules such as acetylcholine. The synaptic cleft is a fluid-filled gap between the axon terminal and the dendron of another neuron. So there is no protoplasmic continuity between neurons. When an impulse or action potential arrives at the axon terminal, it stimulates the movement of the synaptic vesicles towards the presynaptic membrane. Synaptic vesicles fuse with the plasma membrane and release their neurotransmitters into the synaptic cleft. The neurotransmitters thus released bind to their specific chemoreceptors present on the postsynaptic membrane of the dendron. This binding opens sodium-ion channels that allow the entry of ions to generate a new potential in the postsynaptic neuron. This may lead to the development of an excitatory new potential or an inhibitory new potential. Electrical synapses are the less common of the two types. They were first found in crayfish, later in coelenterids, annelids, mollusks.
So in these organisms, you will find electrical
And fish.
synapse.
The membranes of pre- and postsynaptic neurons of electrical synapses are in very close proximity. This allows the direct
Just
flow
like
of
this, they will
electric current
fasten
from
very
one neuron to
fastly.
another.
Even though electrical synapses are very faster, it is not
Synaptic
found in the human.
vesicles
It's
are
very
absent,
rare in the
and
human
only a few
organisms.
mitochondria are present.
Okay? The signals are going to be passing very
Impulse
fast.
transmission across an electrical synapse is faster than that across a chemical synapse.
Clear? So that's all about today's class on the transmission and generation of impulses. We will see with the next video in the next class. That is all about this lecture. Central nervous system and the reflex action. If I get a time today, I'll definitely continue this class today itself. Evening or something. If I get time, I will definitely schedule the class and I'll complete this chapter as soon as possible. Clear? So bye bye guys. See you in the next class with yet another session. Bye bye.